Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you really another awesome guest today involved in creating a better tomorrow for millions of patients out there. Today, we have the honor of being joined by Dr. Oren Gilad, President and Chief Executive Officer of Aprea Therapeutics, clinical stage platform biotech company that's focused on the development of these novel synthetic lethality-based therapies with direct on-target mechanisms of action and clear clinical pathways that target various aspects of the DNA damage response pathways in cancer. Dr. Gilad previously served as president and CEO of Atrin Pharmaceuticals, leading their transaction with Aprea, and he brings extensive experience across multiple phases of drug development. He has previously led development from idea through early stage into clinical development, and as well as securing financing to support all those stages, including regulatory and intellectual property activities. Before founding Atrin in 2011, he authored many high-impact scientific publications over a 13-year academic career, including those that demonstrated the importance of this ATR kinase pathway, which we'll be getting into in cancer development and treatment. Dr. Gilad earned his doctorate in comparative pathology at University of California, Davis, then did his postdoc work down the street here at University of Pennsylvania School of Medicine, and he did his undergraduate bachelor's at the Hebrew University in Jerusalem. A lot to get into, a lot of really interesting themes. Dr. Oren Gilad, thank you so much for taking the time to come on the show today.
Thank you, Ira. Thank you for inviting me and for allowing us to share a prayer with your audience.
I'm looking forward to that, and it's a really exciting set of themes. But before we get there, I'd like to start off as we typically do by just learning a little bit more about you. I take a look back at our guests' writings in the scientific literature. I took a look at your dissertation and was interesting to find out that you didn't start out doing cancer research, but you started work on a sort of tangential area in terms of herpes viruses in various species in fish and chickens, which happen to be oncogenic, and I'm interested in hearing about the connection there. But tell us a little bit, Oren, about the background here before we get to the company and what you're doing now.
Yeah, happy to do so. And you're right. I mean, my journey didn't start with oncology. I actually started with veterinary medicine, where I was the first one to work on this new virus that at the time nobody knew about. It was at the high rate of mortality. The KHV, cold herpes virus. So we coined that term, cold herpes virus. We're the first one to identify the cause of the disease, to identify the virus, characterize the virus. And that was my PhD at UC Davis, and then worked on Merck disease virus, which is also a herpes virus that's causing cancer in chicken. So where viruses and cancer overlap, I think it's what's interesting is that it's the DNA. Because there's hyper DNA replication both in when cell is infected with virus and cancer cell. So that's where I see the homology or the overlapping between viruses and cancer.
And, as we talk about that, and we have, as a couple of months ago, we got our feet wet in the area of the DNA damage response, and talking about BRCA with a colleague down there at Penn, Susan Domchek, and some of the work that's going on there in breast cancer. again, I'd like to sort of go back to that and sort of give you the floor for a little bit just to talk about this area because we forget that, we are sitting here, we are 30 to 50 trillion cells. Every one of them has a DNA rare repair response. When that goes wrong, things go wrong. And, I'd love for you just to talk about DDR for a little bit from a broad context. And then we'll focus in on a little bit about why, and specifically the approach to synthetic lethality, the company's taking, why we approach drugging DDR in certain ways. So please.
Yeah, and so if you really go back a little bit and think about the tumor, what happens inside the tumor, right? So cancer grows. If the cancer doesn't grow, it's not the cancer, right? There's no tumor growth. So in order for that to happen, cells has to duplicate and they have to go through DNA replication. So the DNA has to replicate in order to cells to grow. And that is what we found is the Achilles heel, because that the S phase and the cell cycle, that's the part where the cell is hypersensitive. Double-strand DNA is a very stable molecule, right? Can find in mummies. It survives heat, it survives freeze, it survives everything. When cells undergo S phase DNA replication, that double strand is open up, right? You have the replication fork going both two directions. And single strand DNA is very fragile. It can break. So if you have a fork going bidirectional and you have one break, let's say at 10 o'clock, and another break at 8 o'clock, the DNA is now broken apart. So when we have tens of thousands of replication forks firing at the same time during S phase, the cell has to rely on ATR in the DNA damage and response pathway to make sure that if there is, in case, damage or problem, the cells stop, fixing it, and not carrying the problem through the cell division. So that's the role of DNA damage and response. And what we found that also the Achilles heel of cancer, because when the cancer, and we showed that in one of our publications, let's say under RAS oncogenic stress, the fork will fire prematurely. Normal cell, right, would not enter S phase if the environment is not ready for it, because again, it is a very dangerous place to be. So nucleotide has to be in place. The cell has to be ready to enter S phase. The fork is not going to fire unless it's ready. Under cancer condition, on oncogenic stress, the fork is fired prematurely. So ATR, for example, you mentioned ATR. ATR is hyperactive. And all of a sudden, you have ATR trying to fix the problem, trying to provide the quality control and the service to the cell, and there's just not enough ATR for it. So ATR under certain genetic mutation, under certain replication stress conditions, becoming a target for cancer therapy. And that's what we showed in our paper in 2010. We showed that under certain condition, when we had the experiment, I think you're gonna appreciate it, was a very basic experiment. It was really 4 groups in tissue culture, in cell culture. You had control cells with ATR being reduced, and cancer cell, RAS transformed, with ATR being reduced. Control cells, control ATR reduced. Cancer cell, living normally, everything's fine. Cancer cell with ATR reduced were 100% dead. That was the aha moment, and that's really the scientific experiment that got us to think that ATR can be a good target for cancer therapy. And that's what we've been focusing on, and that's what Atrin was focusing on, and that's what Aprea is now progressing into the clinic.
For the audience, the ATR stands for something a little longer, so ataxia, telangiectasia, and RAD3 related kinase. But in short, it's an important protein kinase that's usually doing its job, sensing DNA damage, activating certain responses. In your particular case, and part of your approach, you view it as it was sort of defined as a compensatory repair gene, and then specifically w- working on inhibiting that uh, within this system of a flawed DNA repair response. And we're talking about a cancer cell now that we don't want to survive, but we want it to enter this disruptive pathway of cell death. In terms of something called synthetic lethality, and I'd like you just to talk a little bit more before we get into the program about what that specifically means.
So, metaphorically speaking, synthetic lethality is as if you see a car driving down the road uncontrolled and you have a blockage at the end. So, synthetic lethality is that something goes wrong in the cell, right? We're not born with cancer. We get 50% of our DNA from our father, we get 50% of our DNA from our mother, and a child is born, right? No cancer. So, something along the way, something goes wrong. Synthetic lethality is really taking advantage of what went wrong. You mentioned BRCA and the work that has been done at Penn. So synthetic lethality actually it's been not just clinically validated, but also commercially validated with PARP. So PARP is synthetic lethal with BRCA loss of function. So the BRCA is the mutation that cause the cells to move from normal to transformed, and PARP is the other pathway that you're inhibiting. So cells, and that's when the two happen in the cancer cell die. Normal cell can live with either BRCA loss of function, obviously because it becomes cancer, or it can also live with just PARP inhibition because it's not right. So, but when the two happens in the cancer cell, that's synthetic lethality. That's where you take advantage of the mutation and you identified what does the cancer cell under these conditions is sensitive to. So, and that's what we're targeting with ATR. There are different mutations. With WEE1, there are also different mutations. And now the field is waiting for the next PARP. What is the next PARP? Is it ATR with ATM? Is it WEE1 with cyclin E overexpression? That's the focus. So understanding the biology, understanding what went wrong, and finding the sensitivity of what we should go after in order to reach that wide therapeutic index by inhibiting a pathway that cancer cell cannot live without and drive it to apoptosis or cell death while keeping normal cell alive.
Appreciate you connecting the BRCA PARP story to this. And again, I think it's rather elegant to, and want to point out for the audience that in this era of sort of systems thinking, systems biology, we are moving beyond sort of the single magic bullet concept and thinking about how all of these, components of a DDR pathway, as an example, have to work together to create these therapeutic interventions. So I think that's important. I'm glad you brought that up. Let's move to the first program, the main lead program, which is focused on ATR inhibition. It is termed ATR 119. Give us a little of the background here. I was reading with interest that it's aside from its mechanism, which you'll talk a little bit more about, it's a macrocyclic drug. So I love natural products and sort of the history of things like rapamycin and the erythromycins and all that. If you can talk a little bit about the compound, nothing obviously proprietary, but and then a little bit about the importance of selectivity here, because this is an area that we have to have a careful balance, because we do need DNA repair responses for the other trillions of cells that aren't cancer. So talk a little bit about the importance of picking a compound that's very selective in this case.
So if I'm missing any of your points, please let me know. But you had lots of components there, and I'm going to address all of them because it's really putting everything in concept and everything together. So just for the audience, and as you said, it's not the magic bullet. This is all about personalized medicine. This is all targeted therapy because it's not chemotherapy. Chemotherapy, we know the side effects, and that's what Aprea is working on. Aprea is focusing on solving the problems that chemotherapy is having. So by matching the disease with the drug, you're really hopefully getting a high therapeutic index when you increase the efficacy and the activity of the drug and you reduce the toxicity. That's the ultimate goal. And you do that by selecting the right patient population to be enrolled on the trial and to be administered with our drugs. How does it work? So and you mentioned also selectivity, and we see that over and over and over again. In DDR, in DNA damage and response, the drug, in my opinion, has to be selective. If you want to do combination therapy, have two selective compounds that you find the right ratio to get the highest synergy, as opposed to a drug that have off-target effects that drives toxicity. When a drug it is selective drug versus drug that has off-target effect, usually you see a higher toxicity associated with the off-target effect because you have to dose up. So you're dosing up your target and you're dosing up the things that you don't want to target. So it doesn't help, it actually takes away from the activity. The reason you mentioned rapamycin and natural products and ATR and microcycle. Let me try and put it all together. When we first started the ATR, so we first on the biology, we identified ATR as a good target. Now, how do you go on the chemistry, right? Because you have biology running on one side, you have chemistry on the other, and the two has to match and meet in order to get the maximum effect. So we went to Mother Nature, and Mother Nature, as you mentioned, selected rapamycin for mTOR, mTOR, ATR, ATM, DNA PK. These are all family members of PIKK. So what we thought said, hey, if Mother Nature already telling us that microcycle is a good approach to selective inhibit, this family member, let's make a microcycle. Let's not make an analog of something toxic and take a shortcut. And that's what we did. And the ATR, there's actually follow-up second generation. And what's nice is that we're seeing a good translation from the preclinical to the clinic. This drug 119 was very safe in preclinical setting. And it's also very safe in the clinic. And we think that the FDA may have also saw that because when we filed our IND, the FDA came to us and say, hey, we want you to reduce the age of patients administered on the study to 12 years and older. And don't worry about any additional tox work. That's FDA coming to us. We went with 18 and older. So that gave us the signal that they like what they saw. And when you have a safe drug that you can treat on a daily basis, and that's another differentiation that we see between us and some of the other ATR inhibitors, that we're treating everyday patient on our trial takes our drug every day. And it's important because going back to the previous question, ATR is active in S phase, right? So cells have to go through DNA replication. So for the tumor to grow, we're assuming one cell cycle per day. So if you're treating every day, you're constantly putting pressure on the cell, on the target, on ATR. If you're forced to treat for 3 days on and then remove the drug for 4 days, it doesn't help, right? Because now you have 4 cell cycles, 4 cycles that ATR is active without being able to inhibit it. So that's another part where we are different than the other ATR inhibitors. And as mentioned, so far, the trial has been moving, and we did announce that we want to also explore BID because it's so well tolerated. We want to give the drug twice a day to try and get a higher target coverage as opposed to just once a day. When you get the coverage, but with twice a day, you can raise the coverage and double it.
So just to sum for the audience at this point, looks safe, well tolerated in humans, no major dose-limiting toxicities. They want you to dose more. I mean, that's another great sign, right? The or one of the components that I noticed on the website was this sort of area of sort of the biomarker component of what you're looking at, and a tool that I believe your group developed known as ReproBiome, which is sort of this proteomic genomic sort of biomarker approach to identifying sort of the right patients, or in this case, you mentioned preventative and predictive medicine here. What exactly is that? And talk, because I have I recently, my father-in-law, recently unfortunately lost his battle with metastatic prostate cancer, but was, as part of his therapy, was given this really amazing sort of oncogenomic laundry list of things going on in his tumor nowadays. And it's kind of interesting where we are with this stuff in 2024. Talk a little bit about how you're using sort of proteogenomic tools in profiling the right patients here.
So first, I'm sorry about your loss. Cancer is a devastating disease, right? Everybody by now has been touched by cancer, and the team at Aprea, we're dedicated to solve this problem, and that's where we spend our days and night trying to cure cancer, and that's our focus. The ReproBiome, you mentioned in the beginning that Aprea also has a platform, and the ReproBiome is very data-rich, a platform that we utilize. When we looked at other platforms like CRISPR, right? Everybody's using CRISPR. CRISPR is a great platform, but CRISPR also has some limitations that directly associated with DNA damage and response, because most of DDR genes are essential genes. They're embryonically lethal, which means that if you knock down this gene, the cell is dead, right? So when you do CRISPR screen and you knock out a gene in DDR, you cannot get any information out of a dead cell. You can be more dead than dead, right? So how do we solve this problem? By going proteomics and overlap it with genomics. So the proteomics, the ReproBiome, is looking at all the factors that are coming on and off the replication fork. Okay, so for example, if you're treating with ATR, how does the cell compensate for the loss of ATR or for the loss of PARP? What the cell has to bring onto the replication fork to overcome the loss of function of a gene or you inhibit. And what it is allowing us, it's allowing us to identify biomarkers, because now you know which factors are coming in. So you want to select patients that do not have functional protein, because then they cannot compensate. It's also allowing us to identify new targets, right? Because it's the same amount of information. And it is also giving us an incline on how cell could potentially develop resistancy. So all of this information is being gathered, and then you overlap it with a genomic, and then you funnel it down to the right mutations. But we are focusing on mutations that are leading to loss of function, not just mutations, because you want to make sure that the mutation is deleterious. It's causing loss of function, and that's our target population.
Nice to sort of see the drug slash the companion diagnostic working hand in hand here. And again, it speaks to the sort of the precision component of what you're up to. So that was very elegant as well. All right, can you talk just? I know the ATRN 119 program is the more advanced, but if you could say a couple words also about WEE1 inhibition, because here slightly different component of the DNA repair response. I was reading something that the sort of cell size and the sort of functionality is an interesting part of what WEE1 normally looks at. But talk a little bit about this and a little bit of how potentially ATRN 119 and this drug, which is also known as APR 1051, can potentially synergize with one another.
Yeah, and we actually showed a good synergy between the combination of ATR inhibition and WEE1 inhibition, RATR and RWE1. And I'll talk about the mechanism there, but before that, need to talk about the mechanism of first the biological function of WEE1. So WEE1 is different than ATR. We talked about ATR being active in S phase, doing DNA replication. WEE1 is active in the transition from G1 to S,
Yep.
G2 to M, right? So oncogene like cyclin E or pushing cells to enter, that's the biomarker and that's the synthetic lethality with WEE1 inhibition. Combination of WEE1 and ATR is that you're allowing the cells to enter prematurely to S phase, and then you inhibit ATR from its functioning in S phase. And that's the synergy. So you let them enter prematurely when they're vulnerable, and you inhibit ATR so it cannot fix the damage for these specific cells under cyclin E overexpression, for example. So that we also we show that, and we're excited to get to the point where we can test it in the clinic. But that's in the future, not right now.
What's happening next? I mean, obviously you have a really innovative molecule with ATR and 119. Things looking great early in the clinic. What's happening next as we get towards the end of 2024 and into 25? I know there's some phase 2 dose work that's going to be happening, but take us through sort of what you envision the next 18, 24 months looking like for this product.
We probably should do a catch up before the next 18 or 24 months, because we're going to be way ahead, hopefully, for where we're now. And we should update your audience, especially when this audience is more scientifically oriented than the business side. So we're expecting good things to come. I'll talk about the ATR is moving into continuing with the dose escalation, and I mentioned BID, so we're going to explore BID as well, in addition to the QD. The WEE1 has two parts to the study. There is the part 1, which is accelerated titration, and then part 2 is a Bayesian design. So we will be advancing. Once we finish the accelerated titration, we will report, do a KOL event, and then move on with the Bayesian design. And there's also the other target in the pipeline, and it's target that we have been working on, which the team is now working on filing the patent for it. And once the patent is filed, we will disclose the target. And I can share that as far as we can tell, this target is not listed on any of the other DDR companies in the field. It's going to be a target that is unique, and it's a target that we have identified through our work, through the Replibiome and through the team. So we're excited about this target to join the pipeline. But we're not disclosing it now. We will disclose as soon as right after we file the patent application.
Excellent. Oren, I mentioned in the bio of the show that you did your postdoc work right down the street here at Penn, and I did not to mention that you're a local sort of Philly, Doylestown ecosystem company. And I just, I'm from Philly. I'm downtown here. I love when, I see companies like this that stay in the Philly area we're becoming known as sort of Silicon Valley, lots going on in ag vaccines and cancer and so forth. Say a couple words just about, how Philly's treated you, how the Philly biotech ecosystem has been growing based on your time and growing your company in it. Say a couple words about that, just for all things positive, Philly.
Well, there's no place like home, and the Philadelphia area is home. The ATR has been born and raised in the Philadelphia area, came from Penn, as you mentioned, to Doylestown, Pennsylvania. The WEE1 has been born and raised in the Philly area. All of the current pipeline in Aprea has born and raised here in Philly. Philly has a lot to offer. I mean, the academic institutions that you mentioned, and also Wharton and other business schools. There's also good seeding money to start companies here, the environment. And so we are the biotech center in Doylestown, and that's a great environment that is supporting innovation, supporting entrepreneurship. They do a great job, providing the infrastructure for companies to succeed. They do a great job providing the infrastructure for companies as they mature. They provide additional support. It's being in the Philly area, and it really helped us a lot. I mean, we are where we are because of right. Part of it is because of the environment that has been supporting the company, supporting our development, also on the financial side, in addition to great talent that we have access to. And it's not easy to not just recruit, but also maintain. It's cheaper than Boston. It's a very vibe. The vibe is very positive, and it's been a blessing being in the Philadelphia area.
Yeah, I get excited when I see companies like yours that gestate and ultimately grow to the stage you're at. So I'm rooting you on in that context as well. Oren, while we have you today, I know you mentioned, sort of, obviously we're both hats. You're focused, intensely on this clinical development, but also financing of the company. I know you get out there sort of on the financial networks and to all those conferences, but what else is happening for the remainder of 2024 as we get into 2025 that we should know about while we have you today? Other conferences in terms whether they're oncology focused, finance focused, anything else happening on the radar, please.
Thank you. And you're right. I mean, we're focusing on spreading the word, and Aprea is publicly traded under the symbol APRE. And I think that also maybe provide an opportunity for your audience to maybe participate and buy some shares in the open market. We are very active in both the scientific conferences and the business conferences. There's a conference tomorrow where we're going to be presenting. We are going to be at the Triple Conference with posters. We're going to make an announcement on that. We're going to share some clinical and preclinical data from both the ATR and the WEE1. So people can follow us, can look online, can follow us on LinkedIn and other media to see where we are and the progress that is being made. But it's open label studies, so we constantly updating with the data as it comes.
Excellent. Yeah, I'm rooting you on. It's oncology, it's precision medicine, it's Philly. I love the story. I look forward to doing an update potentially next year and seeing where things are. But really a great story for everybody again that is going to be listening to this particular episode across the various podcast networks or who will be watching on our YouTube channel. Again, you've been spending time with Dr. Oren Gilad, President and Chief Executive Officer, Aprea Therapeutics. Oren, again, I thank you for taking the time out of the schedule to talk to us for a little while about what you're up to. Thanks to everything you do for the millions of cancer patients out there. And as we like to say on our show, thanks for helping to create a better tomorrow for so many people via the type of research you're doing. Really an awesome story.
Thank you. And thank you for allowing us to share Aprea with your audience. And for your question, it was very comprehensive and very detailed. And thank you for it.
Great having you. Really.